Hello, my name is Madison Sant, and I'm from ARUP Laboratories Parasitology Department. This presentation will cover cestodes, or tapeworms. Please fill out the parasitology practice worksheet while listening to the lecture. Feel free to pause the lecture to take your notes. Write down any questions you may have so we can discuss them when we meet in person. If we will not be meeting in person, I would suggest talking to your professor about any questions you may have. We will discuss the following organisms: Taenia saginata, Taenia solium, Diphyllobothrium latum, Hymelepis nana, Dipudding caninum, and Echinococcus species. Taenia saginata, or beef tapeworm, and Taenia solium, or pork tapeworm, cause taniasis. Taniasis is the infection of humans with the adult tapeworm. Eggs or proglottids are passed with feces. Cattle, for Taenia saginata, and pigs, for Taenia solium, become infected by ingesting vegetation contaminated with eggs or proglottids. In the animal's intestine, the oncospheres hatch, invade the intestinal wall, and migrate to the striated muscles, where they develop into cysticerci. Humans become infected by ingesting raw or undercooked infected meat. In the human intestine, the cysticercus develop over 2 months into adult tapeworm. The adult tapeworms attach to the small intestine by their scolex and reside in the small intestine. The adults produce proglottids, which mature, detach from the tapeworm, and are passed in stool. Taenia saginata adults usually have 1,000 to 2,000 proglottids, while Taenia solium adults have an average of 1,000 proglottids. The eggs contained in the proglottids are released after the proglottids are passed with feces. Taniasis is usually asymptomatic, but may cause dizziness, abdominal pain, headaches, and nausea. The eggs of Taenia species are indistinguishable from each other. The eggs measure 30 to 35 microns and are radially striated. The internal oncosphere contains 6 refractile hooks. Eggs of Taenia saginata are only infected to cattle, whereas eggs of Taenia solium can be infected to humans. Proglottids are longer than wide and can be differentiated by the number of primary uterine branches. Taenia solium contains 7 to 13 branches, and Taenia saginata 12 to 30 branches. The scolex of Taenia saginata has 4 large suckers and no rostellum. Adults are 2 to 8 meters. Cysticercosis is a disease associated with the development of larval form, cysticercus, of the pork tapeworm. Pigs are the usual intermediate host for Taenia solium, but humans, the usual definitive host, can serve as an accidental intermediate host following ingestion of infective eggs. Note that cysticercosis is only acquired from the fecal-oral route, ingestion of eggs, not by the ingestion of cysticerci and undercooked pork, which is associated with taniasis. Humans are usually exposed to eggs by ingestion of food or water contaminated with feces containing these eggs or proglottids. Once eggs or proglottids are ingested, oncospheres hatch in the intestine and invade the intestinal wall, into the bloodstream, and migrate to multiple tissue and organs, where they mature into cysticerci. Some cysticerci migrate to the central nervous system, causing serious sequelae or neurocysticercosis. For review, the eggs of Taenia saginata and Taenia solium are morphologically the same. The eggs measure 30 to 35 microns and are radially striated. The internal oncosphere contains 6 refractile hooks. Taenia solium proglottids contain 7 to 13 branches. Taenia solium adults are 2 to 7 meters. The scolex of Taenia solium contain 4 large suckers and a rostellum containing 2 rows of hooks. Diphyllobothrium latum is also known as fish tapeworm. The eggs are passed in feces into water and yield oncospheres, which develop into chorocydia. After ingestion by a crustacean, the chorocydia develop into porosarcoid larvae. Porosarcoid larvae are released from the crustacean when eaten by a second intermediate host, usually a small fish, and where they develop into pleurosarcoid larvae, which is the infective stage for the definitive host. Because humans do not generally eat these small fish species raw, the second intermediate host probably does not represent an important cause of human infection. However, these small second intermediate hosts can be eaten by larger predator species that can then be eaten raw by human hosts. The pleurosarcoid larvae then develop into adult tapeworms in the small intestine. Diphyllobothrium eggs are oval and are 55 to 75 microns. There is an operculum at one end and a small knob at the opposite end. Proglottids are broader than long, with a single genital port that opens in the middle of the ventral surface. The ovaries create rosette-shaped pattern in the center. Adults are large tapeworms and can reach 2 to 15 meters. The scolex has 2 grooves. Hymelopus nana, or dwarf tapeworm, is found worldwide and is more commonly found in children and institutionalized groups. When eggs are ingested by an arthropod intermediate host, they develop into cysticercoids, which can infect humans or rodents upon ingestion and develop into adults in the small intestine. When eggs are ingested, the oncospheres containing the eggs are released. The oncospheres penetrate the intestinal villus and develop into cysticercoid larvae. Upon rupture of the villus, the cysticercoids return to intestinal lumen, evaginate their scolioces, attach to the intestinal mucosa, and develop into adults that reside in the ileal portion of the small intestine, producing proglottids. The eggs are passed in the stool when released from proglottids through the genital atrium, or when proglottids disintegrate in the small intestine. An alternate mode of infection consists of internal autoinfection, where the eggs release their embryo, which penetrates the villus, continuing the life cycle. The eggs are oval and have a size range of 30 to 50 microns. On the inner membrane are 2 poles, from which 4 to 8 polar filaments spread out between the 2 membranes. The oncospheres have 6 hooks. The proglottids overlap, and adults measure 15 to 40 millimeters. The scolex has 4 suckers and a hooked rostellum. Diapodium caninum is a common tapeworm of dogs and cats, but is occasionally found in humans. It has many common names, including flea tapeworm, cucumber tapeworm, and double-pored tapeworm. Proglottids are passed intact in the feces. In the environment, the proglottids disintegrate and release egg packets, which are also occasionally found free in stool. The intermediate host, which is often larval stages of the cat or dog flea, ingests the egg packets, and the oncosphere within is released into the larval flea's intestine. The oncospheres penetrate the intestinal wall, invade the insect's body cavity, and develop into cysticercoid. The cysticercoid remains in the flea as it matures from a larvae into an adult. The human or cat and dog hosts become infected by ingesting the adult flea containing the cysticercoid. In the small intestine, the cysticercoid develops into the adult tapeworm. Children are most frequently infected, possibly due to close contact with flea-infested pets. Diapodium caninum eggs are round to oval and are about 31 to 50 microns. The eggs contain an oncosphere that has 6 hooklets. The egg packets are about 120 microns and contain 5 to 15 eggs each. Their proglottids average 12 millimeters and have 2 genital pores. They are pumpkin seed shaped and when passed often resemble rice grains when dried. Diapodium caninum adults measure 10 to 70 centimeters long. As proglottids mature, they break off. Hydatid disease is caused by the larval stages of the tapeworms of the genus Echinococcus, and is more commonly found in sheep-raising areas. Echinococcus granulosus causes a cystic form, Echinococcus multiocularis causes an alveolar form, and Echinococcus weigeli causes a polycystic form. Proglottids release eggs that are passed in stool. After ingestion by the intermediate host, a sheep or a goat, eggs hatch in the small intestine and release 6 hooked oncospheres that penetrate the intestinal wall and migrate through the circulatory system into various organs, especially the liver and lungs. In these organs, the oncospheres develop into a thick-walled hydatid cyst that enlarges gradually, producing protoscolices and daughter cysts that fill the cyst interior. The definitive hosts, the dog or cat, become infected by ingesting the cyst-containing organs on the infected intermediate host. After ingestion, the protoscolices evaginate, attach to the intestinal mucosa, and develop into adult stages. Humans are the intermediate hosts and become infected by ingesting eggs. Oncospheres are released in the intestine, and hydatid cysts develop in a variety of organs. If the cyst ruptures, the liberated protoscolices may create secondary cysts and other sites within the body. An ova and parasite exam may be performed on the hydatid cyst fluid. The hydatid cyst will contain a mixture of protoscolices, hooklets, and corpuscles that can be seen in a wet mount. Hooklets can be seen in the trichrome stain. Adults are 1.2 to 7 millimeters, but are not expected to be found in a human host. Several of the images and life cycles are courtesy of the Centers for Disease Control and its Laboratory Diagnostic Assistance website, DPDX. Other images I have taken while working on the bench at ARUP laboratories.